Are you able to hear me? Is it visible? Okay, it is. Okay. So, for in biotechnology, just we are revising the aspects what we covered in biotechnology. And yesterday we covered up to what it is recombinant vector. Gene encoding for antibiotic resistance was cut out from the plasmid of Salmonella by using restriction endonucleases, what we call it as molecular scalpels, by breaking which type of the bond? Phosphodiester bond, which are backbones in DNA. The gene encoding for resistance, that fragment, was introduced into the native plasmid of Salmonella. Whatever the enzyme used to cut the plasmid of Salmonella, the same enzyme used to cleave the plasmid of Salmonella and inserted in the native plasmid of Salmonella by using DNA ligases. What do you call the DNA ligases? It is molecular glues. So, now the vector DNA inserted with the foreign gene, that is foreign gene is gene encoding for antibiotic resistance. Now we are calling it as what it is recombinant DNA. Now this recombinant DNA is transferred to the host cell like it is the bacteria, that is E. coli, similar to Salmonella. How E. coli is similar to Salmonella? Both E. coli and Salmonella both are what it is prokaryotes. Okay. And E. coli provides what? Deoxynucleoside, DNA polymerases. So, what do you call this? Cloning of antibiotic resistant genes in E. coli. We are going to get multiple copies of what it is, the genes. And to modify a genetic system, how many basic steps are there? 3 basic steps are there. Identify DNA with the desired gene, introduced into organism, maintained in organism, and not only maintained in organism, it should be transferred to what it is, subsequent what it is, host cells. Okay. And in this first various tools which are required in order to get success in recombinant DNA technology, we require what? Restriction endonucleases. Other it is DNA polymerases, DNA ligases, vectors, and host cell, which are all required tools in recombinant DNA technology. The first one we require it is restriction enzyme. How the name attained restriction enzyme? In E. coli, as a part of the defense system against viral multiplication, there is a modification enzyme system in E. coli, because E. coli having the prokaryotic bacteria having restriction endonucleases. Why? How the host DNA got protected from restriction, its own restriction endonucleases. So it got what it is protected. And other the enzyme, what do you call it is restriction endonucleases. That activity is to cleave the incoming viral DNA. whatever the enzyme which is cleaving incoming viral DNA and prevent viral multiplication, what do you call restriction endonucleases. Very key point for sake of MCQ. Why the name attained? How it is attained? Because it restricts the multiplication of viruses. Next question. How host DNA got protected from its own restriction endonucleases? The host DNA got methylated. Methylated DNA not recognized by restriction endonucleases. As incoming viral DNA, it is not methylated. It is cleaved by restriction endonucleases. Okay, it is. And number of restriction enzymes so far discovered is 900. From 230 strains of, or it is the prokaryotic, it is bacteria. And first restriction enzyme discovered was HIND2, Haemophilus influenza. It has a particular recognition sequence. What do you call it is? GTC. So G, C, T, A, C, G. It breaks between what it is C and G. 5335. Which ends are found? Blunt ends, otherwise it is non-sticky ends, otherwise what we call flush end in that paragraph information. Very few key points in that paragraph information. HIND2, first enzyme extracted, and it was characterized later, that is 5 years after its discovery. And it has recognition sequence, how many base pair? 6 base pair. Okay. Resulted fragments which are blunt ends, non-sticky and flush ends. Okay, in that HIND2. In next paragraph information, restriction endonucleases belongs to large class of enzyme. What are large class of enzyme? Nucleases. Nucleases belongs to which class of enzyme? Hydrolases according to IUB system of nomenclature. And these nucleases are how many types? 2 types. Exonucleases, endonucleases. As the word itself conveying its an exonucleases. The nucleases act on outside the DNA, ends of the DNA, free ends of the DNA, made into fragment. Exonucleases. Endonucleases, which cut within the DNA at particular sequence. Endonucleases. For these restriction endonucleases, inspect the length of the DNA first and find recognition sequence, that is palindromic sequence, binds to it and cleave the DNA in a particular fashion. E core 1, Escherichia coli RY13 is a strain. E. coli RY13 is a strain. E. coli RY13 is a strain of bacteria. From this, what is extracted? E. core 1. What is extracted? It is E. core 1. It is polypeptide. So, E. core 1 has a recognition sequence. What it is? GAA-TTC. GAA-TTC. CTT-AAG. GCAT-AT. It is 5, 3, 3, 5. Okay. In these bonds, how many total hydrogen bond? It is 14. How many triple bond? 2. How many double bond? 1, 2, 3, 4. How many purines? 1, 2, 1, 2, 3, 3. 6 purine, 6 pyrimidines. Okay. And it breaks between G and A. That is purine, 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 5-3 direction. Palindromic in forward, GAA, TTC. Orientation of reading kept the same, 5-3, GAA, TTC. Okay, it is palindromic sequence. So in this linear fragment, the principality is n plus 1. So if it is 4 cleavage points are there, 5 fragments we are going to get. Okay. And it is circular. N is equal to n. And in this circular, all fragments are going to be like this. It is the basic point in a circular plasmid. N is equal to n. And all the fragments are going to be like this. Single strand at the end. At the end of what it is, double strand on either side. In case of linear, n is equal to n. Suppose 4 cleavage point, 4 fragments are formed. These 4 fragments are going to be like this. But in this, suppose 5 fragments are formed. The 2 fragments are going to be single strand at the end of the double strand. Remaining 3 fragments are going to be like this in case of it is linear. Why exonuclease do not act on circular plasmid? Because it is circular, not having free ends. Okay. In this case, the restriction enzyme. So, symmetric cleavage of phosphodiester bond on both strand at different location between same nitrogen bases away from what it is palindromic sequence. What do you call it is? Cuts, it is staggered cut. The staggered cut are due to palindromic sequence. Single stranded sticky end, overhanging ends which are formed. Okay. Suppose 5, 3, 3, 5. Write sequence on both strands. How can you write this palindromic sequence? On the same strand, C, G, A, T, T, A, G, T, A, T, T, C, C, A, T, A, A, G, C, G, A, it is T, T, A. Now G, T, A, T, A, C, C, A, T, A, T, G, C, A, T, A, T, G. From 3 end, G, T, A, T, A, C, G, T, A, T, A, C. You can write like this. So C, G, A, T, T, A. In the same fragment, it should be like this. You can frame a palindromic sequence like this. Have you got it? Okay with regarding palindrome? Now, and by using agarogel electrophoresis, it was made into fragment. Agarogel is providing saving effect and negatively charged move towards for it is anode. Smaller the fragment, farther away from the gel well and not visible under visible light, treated with ethidium bromide and exposed to ultraviolet rays. Bright orange colored bands of the DNA can be seen and It was taken from each piece of what it is agarogel, what do you call it is elution. The eluted DNA ready for linking with what it is vector. Same restriction enzyme used in case of the source and vector, what are formed? Sticky ends which are matched. If you use this different restriction enzyme, sticky ends are found but it is unmatched. Okay, it is. And with regard to this second, it is vector. So vector, it is a carrier of foreign DNA to host cells for multiplication. Vector is a carrier of foreign DNA to the host cells for multiplication. It is just like so mosquitoes act as a vector to carry what is malarial parasite. So mosquitoes act as a vector. Here, vector are carrier of the foreign DNA to host cells for multiplication. So vector itself a DNA molecule. It is a DNA molecule. It carries foreign DNA to what it is host cells. And in this, it got inserted with the foreign DNA. Now it is vector DNA. You have to get multiple copies of the foreign DNA. It should be transferred to host cells. And the host cell divide. The host cell divide. During every division, it produces what it is the foreign DNA. So, single copy inserted, we got 1, 2, 3, 4, 5, 6 copies. So, getting indefinite number of copies of the linked gene, what do you call it is gene cloning. So, this vector, what do you call it is cloning vector. What do you call cloning vector? Vector is a DNA molecule carries foreign DNA to the host cell for its multiplication. What do you call the vector? It is cloning vector. So it is vector DNA. In this got inserted the foreign DNA. And this vector DNA, that is nothing but it is plasmid introduced into the cell which multiplies along with the cell, every cell division. Okay, it is. So, cloning vector. Vector is a carrier, vehicle to carry what it is the foreign genes to what it is the host cells. And in this, cloning vector, in this cloning vector. That is vector used to carry foreign DNA for its multiplication in host cells. For its multiplication in host cell. That is getting multiple copies of desired gene called gene cloning. Multiple copies called gene cloning. Suppose the question, desired gene inserted in oocyte, the true or false? Whether true or false it is. Desired gene is inserted in oocyte, origin of replication. Suppose it is the vector. Okay. It is ori. Suppose it is foreign DNA. It is ori. It is foreign DNA. So this foreign DNA linked to, or it is right, inserted in, or that is going to be wrong. Okay, inserted in ori means we are deleting what it is. Deleting what it is ori, inserting it. Suppose it is this ori place. It got inserted. What got deleted? Ori got deleted. It is linked to what it is ori, not inserting in case of ori. Have you got this? Got the point? Have you got the point? This inserted in ori or linked to ori. Linked to ori is right, not inserted in case of ori. Others, the other students, rest of the students have got it. It is not inserted in ori. If it is inserted in ori, what is going to be deleted? Ori is going to be deleted. What is important? Ori it is important because it's the sequence of the nitrogen bases which is helpful for initiating the DNA replication. What do you call the site? It is ori. So it is linked to ori, not inserted in case of it is ori. Okay, it is. And in these vectors, bacteriophages and plasmids. Bacteriophages and plasmids are voltage vectors. If you take bacteriophages, bacteriophages are viruses which infect what it is the bacterial cell. It is having high number per cell. Suppose if you take the bacterial cell, one, two, three, four, five, six, seven, 8. It's 8 number. So if virus made to infect what it is the bacteria, it can produce how many viruses? Suppose it is 8 number of what it is viruses. So bacteriophage is high number per cell and equal to copy number of genome. Whatever the DNA have got the capacity in producing the number of what it is viruses. Suppose foreign DNA linked to phase DNA. Foreign DNA linked to phase DNA. You are going to get how many copies? 8 copies of linked DNA. If you link the foreign DNA to the phase DNA, you are going to get what it is 8 copies. Because it is having the capacity to produce inside the cell, it is 18 number. So, bacteriophages having high number per cell equal to copy number of what it is the genome. So, it is also used as what it is vector. Bacteriophages. The first one. Have you got it? If you link the DNA to the phase DNA, it can multiply and equal to what it is the copy number of what it is the number of what it is the phases. And second one it is plasmids. The plasmids are extra chromosomal. That is extra genomic in addition to what it is the genome. Extra chromosomal, circular, naked, double-stranded DNA. Extra chromosomal, circular, naked, double-stranded DNA replicate independently. Replicate independently inside what it is the host cell. Suppose per bacterial cell, it is main genome. It is having 1 or many, even it is 100 may be present. Plasmid. Suppose if you link the foreign DNA to this plasmid, in this cell how many number it is? 2. We are going to get what it is, the 2 copies of what it is linked DNA every time. So linking foreign DNA to phase DNA or plasmid DNA, which multiply and equal to copy number of what it is the phases or it is the plasmid. So whatever the foreign DNA linked to phase DNA and plasmid DNA, multiply equal to copy number of genome. Suppose the plasmid it is produces 100 per cell. You are going to get what it is 100 copies per what it is the cell. So linking of the foreign DNA to phase DNA or plasmid DNA, it can multiply and produce equal to what it is multiply equal to copy number of the genome. If virus is having 100 copies per cell, we are going to get 100 foreign genes, the desirable gene. If it is plasmid, it is having 200 per cell, we are going to get what it is 200 copies. What do you call it is copy number. So extra chromosomal, circular, naked, double stranded DNA present in addition to what it is the genome. What do you call it is the plasmid. And what is the use of high copy number of genes? What is its use? You are going to get multiple copies of the desired gene. High copy number of the genes means many transcripts, that is mRNA. Many polypeptides and high gene product. That's why you prefer high copy number of what it is the inserted gene. You are going to get what it is number of copies. So you are going to prefer the vector which provides what it is high copy number, because many transcripts are formed, and after transcription many polypeptides are formed. Ultimately you are going to get high the gene product, that is the proteins, whatever the products it may be. And there must be some characters, the features of what it is the vectors. The vectors having certain characters. What are the characters of what it is vectors? First it is what it is origin of replication. The sequences of nitrogen bases in DNA where DNA replication is initiated, what do you call it is ori. So foreign gene is not inserted in ori, it is linked to what it is ori. So ori is sequence of nitrogen bases in DNA. Sequence of nitrogen bases in DNA at which DNA replication initiated. Undoubtedly, total DNA is going to be replicated. But initiation of replication occurs at a particular point. What do you call it is ORI. Origin of what it is replication. Origin of replication. So, and next point, you should provide varieties high copy number. Origin of replication is responsible for varieties high copy number of linked gene. Suppose you are linking a gene, you have to get more number of the copies. It must have the capacity to produce more number of the copies. We are going to prefer the vector, the ORI, it should support varieties high copy number. So, ORI responsible for high copy number of linked genes. So, we prefer the ORI with high copy number only, it is preferred. So, prefer vector with a high copy number. So, which is responsible for copy number of linked genes? ORI is responsible for copy number of linked gene. So, by selecting the vector, you should prefer to select the vector with high copy number only, it is selected. Okay, it is ORI, origin of replication. Okay, it is origin of replication. With regarding the origin of replication, okay, it is the sequence of nitrogen bases at which DNA replication it is initiated. What do you call it is origin of replication. So, foreign gene is not inserted in ORI. It is linked to what it is ORI. If it is inserted in ORI, which is going to be deleted, ORI is going to be deleted. Okay. So, that is the first character ORI. And second, it is molecular weight. Suppose it is a bacterial cell. It is the plasmid. You are going to use it as a vector. It should be with the low molecular weight because it has to replicate and it should not be responsible for great metabolic burden on the host cell. If it is having high kbp, more number of nitrogen bases, it requires more number of nitrogen bases. We are going to link a foreign DNA of particular number of nitrogen bases. So, in order to replicate in host cell, it should be with a low molecular weight to reduce the metabolic burden on what it is host cell. So, low molecular weight in order to replicate in host cell to reduce metabolic burden on host cell. Suppose it is having many number of nitrogen bases. The E. coli has to provide many number of nitrogen bases. It has to provide the proteins because Everything is energy expensive process. So energy has to be saved. So it should be with a low molecular weight. The second aspect, Uri and molecular weight. And cloning sites. The third aspect, it is cloning site. Suppose it is the vector. Suppose A, B, C, which are restriction enzymes. So, suppose it is 1 point. So, A is going to act on this point, B is going to act on this point, C is going to act on this point. So, each restriction enzyme at least have one cloning site, one restriction site. Okay. So, each restriction enzyme, it should preferably have one cloning site. One cloning site. Then no complications in case of the further process. Suppose A itself is going to have 1, 2, 3, 4 points. One restriction enzyme having many points, total plasmid is going to be cleaved. So, some useful points are going to be eliminated, making the process highly complex. So, it should have, A should have, it is 1, B 1, C 1, it is feasible, it is convenient. If A itself having 1, 2, 3, more than it is one side, it made into fragment, some useful points which are eliminated, making the process highly complex. Are you getting it? A restriction enzyme having 1, 2, 3, 4 sites, 4 cloning sites. If one restriction enzyme having more than one cloning site, the plasmid made into many fragments. Some useful points which are eliminated. Some having more than one site, make process complex. That is the first one. And some useful points are eliminated. Some useful points eliminated. To get the concept of this, whatever the question it is going to be. Have you got it? With regard to cloning site, okay, each restriction enzyme should have preferably one cloning site, then it is feasible. If it is having more than one cloning site, made into fragments, some useful points which are eliminated. Suppose A, B, C, D, E, F, so many restriction enzyme. But E should have what it is only one side. A should have only one, not it is more than one. B should have what it is one, not it is more than one. Many restriction enzyme can act, but every restriction enzyme should have only one cloning site. Okay, it is. Okay. And next aspect it is antibiotic resistant genes. You are going to get a clear idea about these antibiotic resistant genes at the end of what it is almost the topic. But here we have to know about the layman's idea about what is antibiotic resistant genes. Suppose it is the plasmid. It is not antibiotic producing, it is antibiotic resistant genes. Suppose it is a gene in the plasmid. It is for tetracycline, it is for ampicillin. Tetracycline, ampicillin. And suppose if you clone a foreign DNA, desired gene. So, if you clone a desired gene into a vector, suppose it is a desired gene, it is ampicillin, tetracycline, it is ampicillin. Okay. And other vector, if you take an imaginary diagram, you have to get it. Okay. How can you differentiate the two vectors? In one vector with what it is foreign gene, other vector it is without foreign gene. So there are colony of what it is the cells. How can you identify it is which is having what it is the desired gene, which is not having the desired gene? Okay. So that's why you should have what it is ampicillin and tetracycline, that is antibiotic resistant gene. Suppose it is made to transform into the cells, host cell. Suppose these are vectors. See three kinds of the cells. Okay. So one vector with foreign gene, other vector it is without foreign gene, which are made to allowed into the cells, the transformation into the host cell. We see this cell. So if you take A, B, C, in A, B, C cells, which are said to be transformed in A, B, C, which are transformed. A, B, C, which are transformed. In ABC, which are transformed. Which are transformed. Yes, A and B which are transformed, and it is non-transformed, which is transformed. So it can be identified by using what it is antibiotic resistance. Suppose in this medium there is antibiotics which can survive, only transformed cells can survive, non-transformed cells cannot survive because it is not having the plasmid with antibiotic resistant gene. So antibiotic resistant gene helps in the selection of transformed cells from non-transformed cells. It is non-transformed. In A and B, in A and B, recombinant transformed. In A, B, recombinant transformed. A or B? Yes, A is recombinant transformed. So in this A, recombinant transformed. C, it is non-transformed. You got this? Okay, it is. So antibiotic resistant gene helps in the selection of transformed cells to eliminate from what it is non-transformed cells. What is required? Antibiotic resistant genes which are required. Because if we want to ligate a segment of the DNA into the vector, not all vectors is going to accept this foreign gene. And it made to transform to the host cell, not all host cells are going to accept this what it is this. So three types of the cells are formed. The two which are transformed, one it is non-transformed. And this in these two transformed cells, one is only recombinant transformed. So first we have to identify which is transformed cell, which is non-transformed cell. Then after this, which is recombinant transformed. Okay, you got the idea? So the characters of the vectors are completed. Ori and an antibiotic resistant gene, many antibiotics, ampicillin, tetracycline, chloramphenicol, and kanamycin, which are all normal bacteria, do not possess antibiotic resistant gene. It should be restructured to make artificial vector. It is resistant to what type of, or it is antibiotics. Okay.